So crossing over is the first event that leads to genetic variation in the sperm and the egg, and as a result causes genetic variation in the living organisms that undergo sexual reproduction, such as humans. Now, what is the second event that happens in Meiosis One, in the first division? It's the independent assortment. Assortment means arrangement or organization. So, during independent assortment, the chromosomes, during metaphase, when they want to separate, they separate in a random fashion. Remember that we are speaking about 46 chromosomes, separating 23 to one cell and 23 to the other cell. The separation, it has no rule. Unlike mitosis, the chromosomes at one side will stay at one side and the chromosomes at the other side will move to one side. No. The separation in meiosis, it happens in a random fashion. And there is infinite possibilities of how this separation is going to happen. And again, independent assortment that happens during anaphase 1 in the first division during meiosis contribute as well to the genetic variation that is one of two, in addition to crossing over, that leads to changes among living organisms that undergo sexual reproduction. So if you look at this image, you can see here, we are speaking only about 3 chromosomes. These 3 chromosomes have 4 different ways of separation. Okay, if you check the color, I can make 4 different ways of separation of chromosomes during anaphase. Imagine we have 46 chromosomes. How many possible divisions we might have? It's a lot. So crossing over and independent assortment are 2 events that lead to genetic variation, and they happen during the first stage of meiosis. So this is an overall look on meiosis. It's, as we said, divided into 2 stages, the Meiosis 1 and Meiosis 2. Meiosis 1 includes prophase, metaphase, anaphase, and telophase, and this division is called reductional division because it allows for the reduction of the number of chromosomes in each daughter cell. As a result, at the end of this cell, at the end of this division, I'm going to have 2 cells, each having 23 chromosomes. The second division is Meiosis 2. Also, we call it the equational division, and Meiosis 2 resembles mitosis, where the 2 daughter cells, each one having 23, will undergo 1 more division, resulting in 4 cells. Okay? And again, here we have prophase, metaphase, anaphase, and telophase. It's very important to know that the way meiosis starts is the same as mitosis starts. What I mean is, there is interphase here and there. So, germ cells must undergo interphase at the beginning in the gonads or the reproductive organs. They have to undergo G1 as G2. DNA must be copied. They have to prepare, correct mistakes. All the steps during interphase must be done. Now, after that, meiosis will start in a prophase 1, metaphase 1, anaphase 1, telophase 1. After that, directly it will move to meiosis 2 with no interphase. So there is 1 interphase and 2 divisions. As a result, I will have 4 daughter cells. In order to understand meiosis, we have to look at each phase alone. Starting with the prophase, in prophase there is binding of chromosomes that doesn't happen in mitosis, called synapses. Synapses means the paternal and maternal chromosomes, they line up together as homologs. This image is not common in mitosis. In mitosis, chromosomes, they don't match together or they don't pair together. This pairing will cause the formation of something called tetrad. Tetrad is having 4 chromatids beside each other. I mean, 1 chromosome and 2 chromosomes, each having 2 chromatids, so 4 chromatids beside each other. This is important. Why? It's important in order for crossing over to happen. So for crossing over to happen, the chromosomes must be very close to each other. For this reason, this step happens during Prophase 1, which is the first phase in meiosis. And this is called synapses of chromosomes. As a result, they will be able to undergo crossing over. Now, after crossing over is done, other events in Prophase 1 are similar to mitosis, where the nuclear membrane disappears, the spindle fibers are formed from the centrioles. Then we move to the second phase, which is metaphase 1. During metaphase 1, the chromosomes line up in the middle, but they don't line beside each other, they line up above each other. What I mean is, after the formation of the tetrad, or the 4 chromatids, or the 2 homologous chromosomes together, they will line up as pairs above each other in the equatorial plate or in the center of the cell. For this reason, during anaphase, the separation will be for the chromosomes, not for the chromatids. As a result, in this example, we have 6 chromosomes in the cell. 3 chromosomes will move to one side, and other 3 will move to the other side. And again, remember, during anaphase 1, independent assortment takes place. So the division of the chromosomes is very random, and it's not based on a constant or certain rule. So the paternal and the maternal chromosomes will be separated in a mixed version. I might have in one cell more paternal chromosomes, and the other cell more maternal chromosomes. And this will justify the fact that sometimes you have more traits common between you and your father, and sometimes you have more traits common between you and your mother, because the sperms can carry mixed information. It's not exactly half, 50%, 50% from your father, due to the fact of we have crossing over and we have independent assortment. But when we mean half, the number of chromosomes, yes, it's 23 from here, 23 from there, but these chromosomes might be recombinant chromosomes. It means the same chromosome that you are getting from your father is having a segment from your grandfather, a segment from your grandmother. So there is a combination between the information. So by the end of meiosis 1, I'm having, after telophase and cytokinesis, 2 cells, each one having 1 N chromosome. What I mean by 1 N chromosome, it means 23 chromosomes. Okay? Now after that, meiosis 2 will happen directly after meiosis 1. And meiosis 2, as we said earlier, it's similar to mitosis. So these 2 cells now, each cell carrying 23 chromosomes, are going to undergo prophase, metaphase, anaphase, telophase. The chromosomes will line up in the middle. Chromatids will separate, and they will result with 4 cells. So this is meiosis 2, prophase 2, metaphase 2, anaphase 2, and telophase 2. And again, if you look at these events, you will remember mitosis. The difference between meiosis 1 and 2 is very clear, where during metaphase 1, in meiosis 1, chromosomes line up as pairs, and in anaphase 1, chromosomes separate. Whereas in meiosis 2, chromosomes line up beside each other and chromatids separate. So the number of chromosomes in prophase 2 is 3, and in telophase 2 and cytokinesis, the number will remain 3 in this example. In our body, it's going to be 23. So meiosis 1, reductional division, it reduces the number of chromosomes. Meiosis 2, it keeps the number as it is, but it will form 4 cells, each cell having single chromatid, or 23 single chromatids. As a result, I'm having 4 haploid cells with a 23 number of chromosomes. Each chromosome has single chromatid. Now, this graph explains the content of DNA per division or per meiosis. If we look at this graph, we can see 1, 2, 3, 5, 4, and 5. We can see 3 different values for DNA. Number 1, it's the content or the amount of DNA in G1 before DNA is copied. 2 is the content of DNA after S phase during interphase, when DNA is copied. So we have double the amount of DNA. 3 It's the first division when Meiosis One splits this information, the chromosomes, into two cells, and during five actually it's Meiosis Two, when sister chromatids separate during Meiosis Two. So if I start with a number of chromosomes in one, I will end up having half of it in 4. It means if I have 46, I will have 23. Now, this is the graph for meiosis. The graph for mitosis will be different because mitosis will end up having the same amount, same content of DNA at the beginning and at the end of the division. So, in this model, we can see the different phases of meiosis, and each phase it has its obvious events happening in each one of these phases. At the end, we are forming here four cells that are haploid, each cell having half number of chromosomes. It means 23 chromosomes. Okay, again, meiosis, it's the cell division that occurs in gametes or sex cells. It will result in the reduction of the number of chromosomes. It will result in genetic variation due to crossing over and due to independent assortment. Without meiosis, the continuation of species won't be able to happen, because to maintain the number of chromosomes in a species, the sperm must carry half the number of chromosomes and the egg must carry half the number of chromosomes. In addition to that, these cells that are produced, the sperm or the egg, they will carry each time different information, different genetic makeup. That's it for meiosis. Thank you for watching.